अस्सलाम वालेकुम, वेलकम टू माय चैनल। आज जो हमारा प्रैक्टिकल है वो है प्रिपरेशन ऑफ स्टेन्ड टेम्पररी माउंट ऑफ ह्यूमन चीक सेल्स। थ्योरी: whole mount means the preparation of an entire organism or small piece of tissue without sectioning. For microscope examination, whole mount staining is the staining of entire organism or small piece of tissue without sectioning. For example, staining of cheek cells. Methylene blue stain will be used. Methylene blue stain will be used. Methylene blue stain negatively charged molecules in the cell including DNA and RNA. This dye is toxic when ingested and it causes irritation when in contact with skin and eyes. The squamous epithelium cells from the outer epithelial layers of the mouth will be seen under microscope. The small blue dots are bacteria from our teeth and mouth. Materials required for this experiment includes glass microscopic slides, cover slips, paper towels or tissues, methyl blue solution. Mix approximately 1 part of stock solution in 4 parts of water. Plastic dropper and sterile individually packed toothpicks. Now come towards our next page. Here is the specimen and preparation of temporary mount of human cheek cells slide. You can clearly see the cytoplasm, nuclear membrane, cell membrane and nucleus of the cell which are shown in this diagram. You can draw this on the clear page of your practical. Now come towards our next page. Steps: take clean toothpick and gently scrape the inside of your mouth using side of toothpick. Be sure not damaging the inner lining of your mouth. Point 2. Add a drop of water in the middle of the clean glass slide, then smear the slide toothpick on the outer center of the microscope slide for 2 to 3 seconds. Point 3. Add a methyl blue solution drop and place a cover slip on the top. Remove any excess solution by allowing a paper towel or to touch one side of the slide with cover slip. Place this slide on the microscope with 4x or 10x objectives in a position and find a cell, then view at higher magnification. Hope so this practical will be interesting for you, and keep connected with Vim for more practicals like this.